Okay, hello everyone, and welcome back to the fifth module of the advanced course of practical MR physics for clinician, biologists, and technicians. And I am Sara Bortolani, I'm a clinician, and I will moderate this module. And briefly, I'm a PhD student in neuroscience in Rome, Italy, and I've been studying the field of neuromuscular disease. And with us today is Professor Cornelius Faber, a professor of experimental magnetic resonance at the University of Münster in the Department of Clinical Radiology. And his main area of research is preclinical MR. I just want you to remind that if you have any questions or comments, you can just unmute yourself or use the chat to give questions. So please, Professor Faber.
Okay, Sarah, thank you very much for that kind introduction, and welcome to the audience. I see we have quite a few people, and I would like to emphasize what Sarah already said. So if you have a question, just unmute yourself and interrupt me and ask your question, and we can enter a discussion right away. I think in the end, there will also be some time for discussion. So the module 5 is called Advanced Sequences and Application. And as was mentioned, I'm from the preclinical field. So today you will see some MR of mouse and rat brain, as on this slide here, but of course there will also be a lot of human images. And so to motivate the thing with the topic of today is you all have heard a lot about MR in the recent modules. And I think by now you know that MRI allows for high resolution non-invasive imaging of all organs. And in particular, it's suitable for investigation of brain structure and function. And to look at the brain, 4 specific techniques are quite important or standing out. And this is what we will cover today. So we will talk about possibilities to do structural imaging. We look at susceptibility-weighted imaging, at diffusion-weighted imaging by fiber tracking, as you can see on the top right here. We will talk about perfusion imaging, as indicated by these images, and also into functional imaging. So before we start, I would like to remind you that there are a number of contributions to the MR signal, as you have probably heard during the last modules and also during your basic MR education, I think. So, of course, the MR signal somehow depends on the spin density. If we have no water, there's no MR signal, as long as we do normal MR imaging. But what is determining the contrast is mostly relaxation, at least in most applications. So, you know, we have T1 relaxation, the transverse relaxation, T2, T2 star. But there are other processes like chemical exchange and many other processes we're not going to talk about. So actually what will be the major topics today is the effects of T1 and T2 and T2 star relaxation, and we will exploit them to get information about tissue structure and function. But besides this relaxation phenomena, there's also motion of the spins, motion of the water molecules that influences the contrast. You've probably heard that you can measure flow. This will become important during today's lecture if we look at perfusion. But of course, also diffusion is molecular motion, and this has a severe impact, can have a severe impact if you use the right sequence on your MR signal. And we can exploit the molecular diffusion to elucidate structural details of the object or the subject we are imaging. Okay, and now this is the precise outline for today. As mentioned, we will discuss 4 different techniques where we need advanced sequences, so we can apply the MR method to measure diffusion, perfusion, susceptibility, contrast, and function of, in this case today, the brain. Okay, let's start with diffusion. So what is diffusion? When we talk about diffusion, so diffusion is a very fundamental physical phenomenon. So if you look at a water molecule. So our MR signal is coming from the spins in a water molecule, from the hydrogen nuclei in the water molecule. And this water molecule, of course, if it's not at 0 Kelvin, it will be subject to Brownian motion. So it will move around. And this is shown in this very simple random walk simulation here. So any water molecule, this is just one that is depicted here, will experience a random displacement. And this simulation just simulated here 2,000, 10,000, up to 150,000 of these random displacements, which in our body or in the body of your patient or your subject or your animal come from molecular collisions, which depend on temperature and which are basically random. And now if we look at these steps here, which scale with the time that we look at the diffusion, you see that the longer the diffusion lasts, so the more steps happen, the larger or the area is that the molecule samples. So here it's pretty confined, whereas here it covers some region and it gets bigger and bigger. And this can be described by this Einstein-Smoluchowski relation, and the mean displacement, which is basically the radius of the area covered here, is given by the square root of the diffusion constant times 2 times the diffusion time. So it grows with time, with the square root of time, and also with the diffusion constant. And the diffusion constant is quite important. It's a physical constant. We will come back to that in a minute. Okay, now, how can we measure diffusion with MRI? And this is actually a rather old experiment. It goes back to 1965. It was proposed by Staschiel and Tanner, of course, not as an imaging experiment at that time, but as a spectroscopic experiment. It's also called the pulse gradient spin echo experiment. And what I've plotted here in this pulse diagram is also how we can implement that into a spin echo imaging experiment. You will hopefully recognize that this is a normal, or everything that's in blue here is a normal spin echo experiment, 90-degree excitation pulse, 180-degree refocusing pulse, and we have an echo under read gradient, we have the phase pre-read gradient, the slice selection gradients. Now, what is so special about this Staschiel-Tanner experiment is that we have 2 additional gradients, and these are the diffusion sensitizing gradients. So they are indicated in red here. So they have an amplitude of G, GD, or just G. They have a duration of delta, and they have a separation of here the capital delta. And what this sequence does is special. It basically does nothing to the stationary spins. However, it dephases mobile spins. And this leads to a signal attenuation. We will look at that in detail in the next 2 slides. And what we can use to describe this is the signal attenuation is the so-called B value. We'll also get back to that in a minute. But the B value mathematically can be calculated as this double integral here over this whole sequence duration, so over these 2 gradients. And if these gradients are very short and of large amplitude, then one can calculate the B value as indicated here by basically gamma times the gradient amplitude times delta, everything squared, and then times this delta minus small delta over 3 term. And what you see is that this B value scales with G squared. So the amplitude enters as a square, but delta, so here this is multiplied, delta enters to the 3rd power. So timing is more important than gradient amplitude, just as a little remark. Okay, but let's look why does this provide information about diffusion and how does this work? Again, this is the sequence here. Now our gradients, diffusion sensitizing gradients are blue. And we look at 3 spins. These are the 3 spins here at these 4 time points, A, B, C, and D. So before the first gradient, after the first gradient, and we have the refocusing before, C is before the second gradient, and D is after the second gradient. So right before we start our signal acquisition. Now, these three spins. We look on the magnetization in the transverse plane, the XY plane. After the excitation pulse here at time point A, we have transverse magnetization along the X direction. Everything is in phase. Now, if you apply the gradient, then this is supposed to indicate here the position of the spin. So this one is where there's a negative magnetic field when the gradient is on. So this is supposed to be the gradient here. This is at the center, the isocenter of the gradient. So there is no additional magnetic field because the gradient has the zero. The magnetic field has the zero crossing due to the gradient. And this one experiences a positive magnetic field. So this spin after the gradient at time point B will have a negative phase. This one will have zero phase, and this one will have a positive phase. Now, if we do 180-degree pulse, we flip everything, then we apply the same gradient again. Now, what happens? The same gradient does the same dephasing or rephasing to the spin, and all spins are in phase again. So this is the case for stationary spins. The question is now, why do we do that? We could just leave these gradients out and would get the same result. Of course, for stationary spins, that's true, but not if there's diffusion. If there's diffusion, then these spins move. And we now just look at the left spin. So this left spin is diffusing. It's moving along the gradient direction. So when the rephasing gradient happens here, the spin has diffused a bit, and it sees a different magnetic field, so it's not fully refocused. So the signal is attenuated due to diffusion. This is just another illustration of the same thing here. We have a spin echo sequence. Now I left out all the read phase and slice gradients. We just look at the diffusion gradients. And again, this is an illustration. We look at these 16 spins here. After the gradient has been on, they have a dephasing. So the gradient direction here in this plot is from left to right, perpendicular to the gradient direction. They're all identical. Now, if there's no diffusion, we just flip them back, and the same gradient will refocus them, and we will have the same situation as before. But if there's diffusion, then we can assume that some of these spins here indicated by this yellow arrow exchange their position. So this one will move to this position, these here and these here. And now if we have the refocusing, they will not be in phase again because they experience a different gradient during the rephasing than during the dephasing. So what the resulting magnetization will be, or the situation of the spins, most will be refocused, but some are not. And there are also some spins indicated here that move perpendicular to the gradient, and those, of course, will be rephased because moving perpendicular to the gradient has no effect in its diffusion gradient. Okay, now, what does this mean? So again, the sequence up here, we have the situation where all the magnetization is in phase. If we have no diffusion, we have a normal spin echo. Everything is in phase again. Of course, we have some T2 decay, which is neglected here. But if we have diffusion, then we have additional dephasing due to this pair of diffusion sensitizing gradients. So we have T2 decay and diffusion. Okay, now the signal decays, and as I've mentioned before, this decay depends, of course, on the strength of the gradient and can be expressed by the B value we've seen 3 slides before. Now, if we measure the amplitude of the signal here, then this signal amplitude will be attenuated independent of the B value. So the signal with this sequence will be basically the original signal as 0 with a damping factor, the exponential of minus B times a diffusion constant. And this is not the physical diffusion coefficient. This is very hard to measure, in particular in vivo. So we call this the apparent diffusion constant. So the signal is attenuated by an exponential term with minus B value times the apparent diffusion coefficient. And the apparent diffusion coefficient is, so to say, the diffusion constant that we can measure in vivo. It's not the pure physical constant because there are other processes playing into it. Okay, now, let's look at this diagram here. Styscale and Tanner have proposed this sequence 50 years, no, more than 50 years ago, actually, 57 years ago. And they just did a spectroscopy experiment. So they did no imaging, they just did the diffusion weighting, then acquired a spectrum and measured the signal amplitude. If you want to do imaging, then of course we can do it as indicated here. We can do a spin echo experiment, but of course, then we need to wait a TR and start again and again. And if we want to have a high resolution image, you know, we have to do 200 phase encoding steps, and this will take forever. And this is why practically diffusion measurements are separated into a diffusion preparation and into a fast readout. So usually this thing is done in the beginning, and then we combine it, for example, with a fast echoplanar imaging readout, so a multi-gradient echo. And after the diffusion preparation, the whole image, or in the case of a segmented EPI, part of the image is acquired, and then you need only a few repetition times to come to one repetition time to come up with the full image. This can be a single shot EPI, so reading out all of the data, all of K-space at once. A segmented EPI is like 2, 3, or 4 shots. Or you can apply any other fast sequence depending on what you see. So you could do this with a triple spin echo, or with a balanced sequence, for example. However, EPI is still the fastest and the most robust one. And one remark to the diffusion preparation, what we see here is the so-called pulsed gradient spin echo sequence. Of course, there are other diffusion preparations. There's, for example, a pulsed gradient double spin echo, where just the spin echo with the 2 gradients will be doubled. Or one can have other shapes of gradients, like oscillating gradients or specially shaped gradients. However, this gets a bit complicated, and we're not going to talk about that today. The important thing that you should remember is that we have a diffusion preparation and then a fast readout, which is usually done by an EPI sequence. Okay, now what can we do with that? Of course, we can vary the amplitude of our gradients or the timing, and we can vary the B value. And if we do that, this is shown here. So we have changed the B value as indicated on this axis down here. We've done measurements for 9 B values. We've repeated them 4 times. So this B0 is just a spin echo experiment, and then we've increased the B value, and you see it follows an exponential decay as given by this equation here. We've repeated the measurement 4 times to show that it's very nicely reproducible. However, if we're down here, then we get into the very noisy regime. Of course, this is a signal attenuation, so here's a lot of noise. So here is some noise, and the signal to noise ratio gets low, and the curve, of course, deviates. However, you can do fitting of this curve according to this equation. And since we know the B value, we can obtain the apparent diffusion constant of our tissue. And if we do this in an image, we can do this pixel-wise. And then we know the local diffusion constant of our tissue. And of course, the diffusion contains information about the microstructure. So why is that? Well, at first, we look at the result here. So if we do a diffusion weighted image, like the B use, for example, a high B value of 1,000 seconds per square millimeter, this is an image of a human brain, a human patient that has an acute stroke in that case here. You obtain an image, and what you see here is suddenly bright regions popping up. And this is regions with reduced diffusivity. So there, the stroke is located, the cells are swelling, the water is hindered in diffusion, it cannot diffuse as much, so the signal decay is slower or less pronounced, so the diffusivity is reduced, and this area appears bright in the diffusion weighted images. Now, if we do this for, as shown for 9 or a number of B values, we can calculate this apparent diffusion coefficient, and this is the same brain here, just calculated this apparent diffusion constant here. And here you see the same areas are suddenly dark. So why are they dark? Here, the signal is increased because the diffusivity, so the diffusion constant, is low. And of course, if we have a map, a low value of the diffusion constant in this color scale is a black value. So if you have reduced diffusivity, it's bright in the direct diffusion weighted image, but it's dark in the ADC map. This is the important difference, and not to confound each other. Okay, now, why do we obtain information about microstructure? Well, of course, I said already with cell swelling, so something happens, so the signal changes, but we can also obtain structural information. Now, I brought you here an example with a very high resolution because this illustrates details very nicely, I think. So this is a red spinal cord that has been scanned in a preclinical MR scanner, in vitro in that case. And we do just this diffusion sensitizing gradients along the slice direction. So this means into the plane here, into the screen. So what we see is in the spinal cord, we see here around, we see a rather low signal. So there is high diffusion. And in the gray matter here, in the central butterfly of the spinal cord, we see high signal. So why is that? Of course, the cells, the neurons, the axons go down the spinal cord, so in the direction of the diffusion gradient. So there's a lot of motion into the screen here, but not in the gray matter where the cell bodies are and there's no structure. So we can already estimate or guess from these images there's a structure along the diffusion direction where the signal is lower. So what is quite interesting is that in the white matter we see these 4 stripes here, and also here we see some stripes. The question is, what is going on here? Now let's move to the next image where we apply these gradients along the y direction. So here from top to bottom of the spinal cord sample. So here we see the contrast has basically disappeared. So it's in the gray and white matter. It's basically the same contrast. However, we now see these 4 stripes and also here. So these are projecting nerve bundles that come out of the gray matter and go along that direction. So the diffusion is increased in this arrow direction here. So the attenuation is higher because the water has increased diffusion along this direction of the axons. Let's make the test and do the x direction. And if we do this, gradient diffusion sensitizing in direction, here those stripes have completely vanished and most of the contrast has vanished. So already from these images, we can conclude that there are some microstructures in our tissue, although we may not even be able to resolve them as with this very nice high resolution here, but we can infer on microstructure. Mathematically, if we do this, we need the diffusion tensor, so a 3x3 mathematical object, the diffusion tensor, to describe this mathematically, and we need at least, so it has symmetric, it has 6 components, so we need at least 6 independent measurements, and then we can calculate this diffusion tensor. One remark, in practice, you do much more, many more than 6 directions, usually 30 plus or even 50 or 60 directions if you want to sample all the diffusion directions. So you apply linear combinations of these gradients that we have seen here, yeah? These gradients, any kind of linear combination, and then you can calculate the diffusion tensor. Now, what can we do with the diffusion tensor? We can do some math, do a diagonalization, so calculate the eigenvalues. We can have this diagonal matrix here with these main eigenvalues, the eigenvectors, and we can plot them. And if there's isotropic diffusion, then it's a complete symmetric sphere. If not, it's elongated, like this blimp shape here. And this tells us that there's, for example, in the spinal cord, one predominant diffusion direction. From that, we can calculate the so-called fractional anisotropy. So lambda are these eigenvalues of diffusion. And this fractional anisotropy, if it's anisotropy, if it's isotropic, then it's very low, it's close to 0, because all the eigenvalues are very close to the average, the mean value. But if there's one predominant direction, then the fractional anisotropy is large. Okay, how does this look like here? this is mouse spinal cord actually, but human looks a bit similar with better resolution, of course, in many cases. Here we've calculated the fractional anisotropy. Here there's the gray matter. There is completely isotropic, so very low anisotropy. On the white matter, there's a high anisotropy. And so here are the different contributions. And how this is plotted is usually in this color coding. So blue means that there's a direction into the screen, as in the white matter. And well, in the center, it's black, so there's no preferential direction. And what can we do with that? If we have this diffusion tensor, we know in any pixel, we know the preferential diffusion direction. And then we can like plot these main diffusion direction and the anisotropy. And we could follow the main axis, like going up here or here. And this is most likely the direction where fibers run. So in the rat brain, in that case, we have the corpus callosum. We can follow the fiber from the left to the right side of the brain. And this, of course, also works in humans. And this can be done at very high resolution. And again, here's this color representation. So this color representation shows you blue pixels show a diffusion into the screen, green show from top to bottom, and red from left to right. And again, this is mouse in that case here. We have in the corpus callosum left to right connections. We have some blue directions into the screen and green from top to bottom. Okay, this can also be done in human, like in this patient here, showing where fibers are still intact and where it's massively affected by a lesion. So if you want to apply diffusion tensor imaging or diffusion weighting to human subjects, then you usually use this pulsed gradient spin echo. You usually combine it with a fast readout, the EPI. If you want to resolve microstructures, you have to use many preferentially high B values, and you should use many diffusion encoding directions to come up with this diffusion tensor from which you can calculate your fractional anisotropy. Okay, to sum up the diffusion part. So diffusion weighted imaging and diffusion tensor imaging probes microstructure of tissue. We use the PGSE, pulsed gradient spin echo, for preparation and combine it usually with a fast EPI, echoplanar imaging readout. So the diffusion weighted imaging can provide you with an ADC map, which tells you about tissue alterations like cell swelling. However, if you want to have more detailed information about directional structures, then you need diffusion tensor imaging with 6 or more gradients. Typically use many more. You can calculate this fractional anisotropy, which indicates, for example, white matter alterations, and you can do fiber. Okay, are there any questions regarding the diffusion part?
Well, now I can't see any question from the chat. Maybe we can just wait a bit.
Yeah.
Well, I have just a curiosity, maybe it's so stupid. You said the ADC, we use the ADC, the abbreviation, and the apparent diffusion constant, I didn't get why you call apparent, the meaning.
Yeah, I mean, diffusion is a physical process, but of course, if we do an MRI measurement, we also have T2 effects. We may have chemical exchange effects or magnetization transfer. So other processes may also attenuate our signal, and we have no chance of separating that. We cannot, well, we can be sure that it's not only diffusion, there are other processes. This is why it's called apparent, because this is what we see with the MRI, but it's not truly 100% diffusion, but other effects as well.
Okay.
Okay, any other questions? Then we move on. We're a bit behind the time, but the other parts are shorter, so diffusion was the longest. Let's talk about perfusion. What is perfusion? Perfusion is the term that describes supply of organs with blood. Perfusion is determined by blood flow and, of course, by the properties of blood, and it tells you how much nutrients like oxygen or metabolites are brought into the tissue by blood flow. So when we talk about blood flow, we can, or we want to look at it, we can look at it in a very schematic way. So this blue tube here is supposed to be a blood vessel. And of course, if there's flow, then we have moving spins. And of course, these motion of spins will influence our MR signal. So for example, if you do excitation of a slice or saturation, and we wait a few milliseconds, seconds, then this slice will have, the blood will have moved, and it will be at a different position. And this is what exploited for the time of flight imaging, for example, but this is also what is exploited to measure perfusion. However, there's a second part. This is a very important contribution. This is T1 relaxation. And now this slide has nothing to do with perfusion. It's just to remind you that there's this inversion recovery experiment. So this is just a gradient echo inversion recovery. And if you look at the brain, for example, you know, you've heard, I guess, that the fat is recovering very fast due to short T1, gray and white matter quite slowly, and liquor very slowly. And then you can do this flare sequence where you null the liquor signal. So this is a mouse brain here. The black is the liquor, and it's nulled by this fluid attenuated flare sequence. But this is not useful to measure perfusion. But it's important to remember that we have these different degrees of relaxation and that we can exploit it to alter our signal. So how is perfusion measurement? And there are a number of ways to measure perfusion. Of course, there's some contrast-based. The gold standard is probably using PET, it's radioactive water. But we can do it with MRI, and we have a very convenient sequence, which is called arterial spin labeling, or ASL. And this is a sequence that is contrast-free. That's the great strength. The problem is, however, that it's a bit tricky to obtain good data with it. But the principle is that if we want to image this slice and we want to measure perfusion of this imaging slice in the brain here, we apply a saturation or an inversion slice, which can be at the same position or usually is different. So if you think about that, the blood is coming from the heart, it's flowing up the neck, and you have the saturation slice somewhere down here, you will saturate all the blood before it's reaching the brain, before it's reaching your imaging slice. Now, if you look at after an inversion, then your signal will recover. And if you do an inversion, for example, the T1 relaxation will follow this curve here after an inversion. And if you don't do it, then you will have a control image and you will have a difference. And now if the saturation slice is not at the position at the imaging slice, there will be some time that the blood requires to flow up there, and then this will modify this relaxation. But we will look at that in detail now. Let's assume we have this imaging slice. So we have imaging slice and saturation slice. They don't have to be at the same position. The imaging slice is here. The inversion slice is here. Now, these are our spins, the water spins. They are all relaxed. But if we do an inversion, they are inverted. Now, if there's blood flow into the brain, then at some position, if we keep on inverting, at some point, these inverted spins will come into that slice here, and our signal will be lower. So we can detect that if there's no blood, we will see no effect of a remote inversion slice. If there's blood flow, we will see an effect, and the faster the flow is, the more the effect will be, depending on our timing that we choose. Okay, so we can, if we simply do that, we can obtain information about the perfusion of the tissue, so how much of the saturated blood is flowing. So this method is called the continuous arterial spin labeling because we do inversion all the time. This may be problematic because we need a lot of RF power, and also some other challenges are related to that. This is why people came up with the pulsed arterial spin labeling, and this uses a method that's called FAIR. It's not FLAIR, it's FAIR. It's the flow-sensitive alternating inversion recovery. So what we do is we have an imaging slice, and now we place the inversion slice at the same position as our imaging slice. So what we first do is we look at our spins, and we first do a global inversion. So we invert all the spins. And then we will, after this inversion pulse, we will have a T1 relaxation. And even if we have flow, it will be the same because everything is inverted. And if the spins are inflowing, there will be no change. However, then we do an experiment where we invert only the imaging slice or any kind of slice. And now if we have flow, what then happens is that in addition to the normal T1 relaxation, there will be inflowing, not inverted spins. And this will lead to a faster, apparently faster relaxation. And this difference can be exploited and will tell us about the degree of perfusion. So how does this look if you apply this to a subject or a patient? So this continuous arterial spin labeling, you have the saturation slice separated from the imaging slice. So you can, for example, do this here in the neck where you separate all the carotids. You can place it very close here. But if you use the pulsed arterial spin labeling, it's usually at the same position. And one important thing is usually you choose the inversion slice a bit thicker than the imaging slice to measure your signal changes. Okay, this is from preclinical experiments. This compares in the solid lines, the continuous with the pulsed arterial spin labeling. And you see that the continuous is more efficient and also that at higher magnetic fields, it is higher. But we're not going to go into detail because, well, this requires a lot of math to calculate the signal. I cited some of the literature in the appendix where you can look into detail. So perfusion is the blood, basically describes the blood supply of tissue, and we can measure this by the arterial spin labeling. Arterial spin labeling does not require contrast agents, so we use the spin tagging, or we label the spins to probe the perfusion, and we have this pulse or the continuous version, which both have some advantages and disadvantages. It very much depends on your particular application. But in most clinical scanners, there are sequences which work quite stable. From the preclinical world, unfortunately, in mice and rat, it's very difficult to measure perfusion still today. Okay, are there any questions on this short part of perfusion measurements?
Thank you, Cornelius. It was really interesting, and yeah, I think we all wanted to have more time to go into each part.
Okay, so then we move on.
We have no question right now.
Yeah. Okay, great. Then we move on to the susceptibility weighted imaging. Okay, so susceptibility weighted imaging is another technique that provides information on microstructure. And as it is said, it measures susceptibility effects. And the thing I would like to clarify from the beginning, so SWI, susceptibility weighted imaging, is not a real image, it's not an imaging sequence, it's actually a post-processing technique that exploits magnitude and phase images in susceptibility sensitive sequences. Okay, let's look at that. So what you see on the left-hand side, this is taken from a recent review, also cited in the literature list. You see that something is wrong with the patient here, but well, it's not pretty evident how far this extends over the brain. In the spin echo, it's T2 weighted. If you look at the T2 star, you see something that looks like bleeding, so hemorrhage. And this is why we can apply susceptibility weighted imaging. Here, you suddenly see a lot of bleedings in the brain, which are basically invisible, at least in the spin echo, and hardly visible in the T2 star weighted image. So how does susceptibility weighted imaging work, or what's the idea behind that? And so susceptibility weighted imaging usually uses spoiled multigradient echo sequences. So we acquire, we excite the magnetization and we acquire a long echo train and calculate images. So we can use each echo here to calculate one image. If we, of course, if we repeat that, if we don't do that, I just realized that the blip should be away here. So if we use each echo to acquire an image, then we get these magnitude images which decay with the T2 star constant. However, you have heard that MR signal is a complex number, so it has a magnitude and a phase. Now, if we calculate the according phase images, we see it looks terrible, because with the increasing T2 star effect, we get this huge phase wrapping over the brain. So how can we exploit that? We can actually exploit that. And that's the idea of susceptibility weighted imaging. You have these big phase differences, which gives phase with more than 360 degrees, which then causes artifacts. So you need to do some phase unwrapping. So basically you filter out these large phase variations and you only have the small phase variation. If you do that, you suddenly start to see phase differences similar to the magnitude image, but they are a little different. And from that, you can calculate a phase mask. And if we use this phase signal and we multiply it on the normal image, the magnitude image, usually this is done 4 times, multiplied 4 times on the magnitude image, and we suddenly get a new image, and then we can look at it in a maximum or minimum intensity projection. So how does that look? This is, again, an example. So here, this is a normal gradient echo where you already see a lot of microbleedings. And this is a susceptibility weighted image. And now, if you look closely, here, this white arrow points out a microbleed, which is not visible in the gradient echo image. And now, if you use this and do a minimum intensity projection, then you suddenly get this, and you very clearly see all these microbleeds. So we can use the phase contrast in a T2 star weighted image, the phase information, to enhance this susceptibility effects. So where does this come from? Of course, a very important interaction is the dipolar field interaction. And susceptibility change, this delta chi here, always gives rise to a dipolar field. So the details of this equation are not so important, but any little susceptibility difference gives rise to a little dipolar field, even in the normal tissue, but they're very small. If we have something like a vein, for example, here, we have a bigger dipole field. And if we have here like air tissue interface, we have a huge dipole field. And these give rise to differences in local phase. Here we'll have a huge phase, here we'll have a minor phase, and here we'll have basically a very minor phase change. Now, if we do that, then we can use this for susceptibility weighted imaging. And here's a table that shows you that several diseases on the right column here. This is basically a number of conditions or phenomena or diseases that produce some kind of mostly iron containing, but also just other lipid molecules, calcium containing molecules. And these have, this should be at the chi here. This is the stability difference. This is in CGS units. And you see that some paramagnetic molecules like deoxyhemoglobin, methemoglobin, hemosiderin, ferritin, they have a large paramagnetic susceptibility, which give rise to large effects, which will have large phase differences. And if we detect them with SWI, then we can find hints to, for example, sickle cell disease, hemorrhage, Wilson's disease, or so on, or even demyelination, dysmyelination. If you want to do this practically, so in principle, you don't need a special sequence. You just can run a multi-gradient echo sequence and then do it all on your own. Of course, most vendors have installed ready protocols to do susceptibility weighted imaging. On Siemens, for example, it's called SWI. On the Canon scanners, I just looked that up in the review. I have never been at the Canon scanner. It's the FSBP sequence. On the Philips scanner, you can use the 3D SWI-P sequence, or you can just use the raw data of a gradient echo sequence. On GE, it's called obviously SWAN, and on the United Newsoft, it's also called SWI. We're not going to go too much into detail. There's some further things you may want to consider, but the basic principle is that we have a phase and a magnitude image, and they are multiplied, and this enhances the effect of susceptibility difference, which most importantly give you a hint to iron storing diseases. One important thing, if you do that, is that you should use flow compensation. You've probably heard that flow creates an additional phase. Now, if we make a phase image and we have a phase that's coming from the flow, then this will create artifacts in the SWI. And here we have, I think this is data from a Siemens scanner. Here we have a multi-gradient echo, first echo, second echo. This one is not fully flow compensated. So you see, well, the flow is invisible. Here it's fully flow compensated. This is the SWI image where you can nicely see the veins. And if you look at this, it's the same image, well, the same patient, but without flow compensation. Then these yellow arrows indicate some structures that are not in here. So these are artifacts due to the missing flow compensation. So if you want to do SWI, remember, SWI enhances contrast from susceptibility difference. And it can be done with any T2 star weighted sequence. It's a post-processing which uses repeated multiplication of the phase image onto the magnitude image. It enhances contrast from veins, microbleeds, and other lesions, mostly iron-containing lesions. And very important, you need to do flow compensation. Okay, that was the SWI part. We still have the functional part, which is also not too long, but are there any questions on the SWI?
I think our audience is either very competent or really shy.
Oh, it's going too fast. Okay.
No.
Okay. Yeah, I mean, for the audience, if it's too fast, just interrupt me and ask to explain again. This is not a problem. Okay, we have 10 minutes to go. We do some functional imaging if there are no questions with the SWI. Okay, so functional imaging. MRI can image function. Of course, you can image heart function, you can image motion, but if people talk about fMRI or functional MRI, most commonly people mean measurement of activation or activity of the brain. And this can be done under stimulation or at rest. And what fMRI most often does is it exploits the BOLD effect. And BOLD stands for blood oxygen or blood oxygenation level dependent effect. So what is that? It's a bit complicated, but we will come to the MR in a minute again. Now, we can measure the BOLD effect, but what we want, of course, we want to see our brain work. So if there's a stimulus, for example, if you listen to me talk, your auditory cortex will do some neural activity. And this, in the end, gives rise to BOLD signal. If you would be in the MR scanner, I guess most of you are not right now. However, this connection between the neural activity or response and the BOLD signal is very complicated. It's called the neurovascular coupling and depends on the cerebral blood flow, cerebral blood volume, so flow and volume, and on the oxygen consumption. So why is this measurable? Why can we detect this? And this is a bit similar to susceptibility weighted imaging. You know that our blood is red. So why is it red? Because we have a molecule. This is this protein here, hemoglobin. Hemoglobin transports oxygen through our blood. So if our muscle works or if our brain works, then the blood brings fresh oxygen because the tissue needs oxygen. And this is done by this molecule. This has 4 of these very protoporphyrin, or simply called heme molecules. They are in red here. And this iron can bind to oxygen. And if it's bound oxygen, then it's diamagnetic. It has a negative susceptibility. And if it's deoxygenated, so if there's no oxygen, it becomes suddenly quite strongly paramagnetic. We've seen that in the table of the susceptibility weighted imaging. Yeah? So if there's no oxygen, then it's paramagnetic and it creates some magnetic susceptibility. If it's oxygenated, it's diamagnetic. Now, why does this change signal in the brain when the brain thinks? Well, first we have to talk briefly about how our brain looks. Now, if we could, well, this is from animal tissue, but if we could look in our brain, we would see that there's, of course, we have neurons and there's astrocyte cells, but there's a lot of blood vessels because the blood comes in through the arteries, arterioles, capillaries, and through the veins. And if you would be able to look into the brain, you would see a very dense network of capillaries. So there's basically blood everywhere. But those capillaries are very narrow. You can go down to a few micrometers, so that only an individual red blood cell can squeeze through at once. And these red blood cells, they have a large number of these hemoglobin proteins and of these heme molecules. So there are more than 200 or around 280 million heme molecules per red blood cell. And the point is now that in the capillary bed, the oxygen is diffusing into the tissue where the oxygen is consumed. And this is leading to the fact that the arterial heme is basically saturated, while the venous heme is only saturated by 75%. So 25% is suddenly paramagnetic, and this is paramagnetic effect that changes our MR signal. So this is shown here in one of the seminal papers. So basically, this is a tube of blood, and there are spin echo images done on oxygenated blood and on deoxygenated blood, and a gradient echo. And you see in the oxygenated case, it's the tube is, well, it's dark, but you see the tube. But if it's deoxygenated, then suddenly the field is distorted, and here in the gradient echo, it's massively distorted because we have this T2 star effect due to the paramagnetic effect of the hemoglobin. And this is Seiji Ogawa. It's a very poor image, I have to admit. Published this first in MRM and then in PNAS in 1990. And he measured this in rat, actually. So what we see here is a rat breathing normal oxygen, and you see all the blood vessels. Obviously, because these veins are to some degree deoxygenated. And then he gave the rat pure oxygen to breathe. So much more oxygen that is needed. So all the hemoglobin has been oxygenated, has been diamagnetic, and it's the same image, but the blood vessels have disappeared because this diamagnetic effect it rules out the paramagnetic signal cancellation. And this is illustrated here. So this is supposed to be a blood vessel. If the degree of deoxygenation increases, here indicated by this capital of these large letters, then the field, this is supposed to be the field distortion, causes dephasing. And with low deoxygenation, there's a lot of signal. And with high deoxygenation, is a lot of dephasing, and you suddenly see the blood vessels, or your signal goes down. Now, how can we exploit that? And this is data from the Boas lab from Boston. They have made this computational model. So this is capillary from the cortex. So this is how it looks in your cortex. Well, this is actually from mouse, but in the human, it looks very similar. You have the fresh blood coming in, going through the capillaries, and then flowing out through the veins with reduced oxygen content. And they simulated that, so how over time the fresh blood flows in and would distribute, and how the oxygen would move through the tissue. And what they then did, they calculated here through cross sections, basically the field distortion. So every little capillary makes a magnetic field distortion. Then they assumed the water molecules diffusing through these fields, and of course leading to some dephasing, signal loss. And they calculated for a gradient echo and for a spin echo. And what you see, if you have a stimulus, blood is flowing through a tissue, then you will have some change in the gradient echo sequence, and you will have some change with the spin echo sequence in response to blood flow that is triggered from a stimulus. For example, when you now listen to me, your auditory cortex is doing that all the time. Or if you look at the screen, then your visual cortex is doing that all the time. And this bold effect, so this signal change is called the blood oxygenation level dependent, or bold effect. This, of course, depends on many, many things. Depends on the vessel diameter, on the orientation of the vessel, which sequence you use. It depends on the TE, the echo time you apply, on the magnetic field, on the spatial resolution, and many, many other things. And if you want to measure that, then you can use gradient echo or spin echo sequence. And as a matter of fact, gradient echo sequences are most sensitive because it's a T2 star effect. Further looking into the details, spin echo is not completely useless because it's also more sensitive to the capillaries, not to the large vessels, but usually one uses gradient echo sequences to measure this ball. Okay, now let's briefly come back before we come to the few final slides. So we want to measure the neural response, but we can only measure the bolt signal, and this depends on this very complex neurovascular coupling, which is not fully understood. However, I try to summarize this here. So red is the bolt signal that we measure. If there's a stimulus, then the solid line here is the oxygen consumption. The oxygen consumption goes up, so the deoxygenation will increase here and your signal will go down. But then our brain notices we need oxygen, and increases the blood flow. And then the deoxygenation level goes down and your signal goes up. So the bolt signal is usually positive signal because our brain, our brain starts working or our muscle starts working, then a lot of blood is transported there. So we have during activity, we usually in the brain, we have more oxygen than without activity. And this is what is the bolt. Now, how can we measure that? If you look at this oxygenated blood, it decays, and deoxygenated blood decays a bit faster. So there's a difference. And now we can modulate, for example, the blood oxygenation by breathing air and oxygen or air and carbon here. So if we have the slower decay, we will have a higher signal in our gradient echo images. And if we have the faster decay, we will have a lower signal. And then we can, of course, we can do this pixel-wise for any pixel, and we can do a statistical test, and then we can obtain information where in the brain this change takes place. Yeah, let's jump across that. And the second last slide, I think. If you want to probe this in the brain, then you usually present a stimulus. So this again is a red brain here. You give a stimulus and you measure, give a stimulus again and again, and this is also shown here. So you do this repeatedly, and then if you do this 3 times, the green, blue, and red, you always see a similar response, and then you can do some statistical testing and see where in the brain the signal follows your stimulus pattern. And for example, if you would be in the MR scanner now and would do an MRI scan, you would see that your auditory cortex is active because you listen to me. And probably if you look at the screen, also your visual cortex would be active. And the statistical testing would indicate that every time I start speaking or change the slide here, then there is some activation that would be detected in your brain. And you probably have heard that there's also resting state fMRI. This is just a different statistical analysis, but due to the sake of time, we're not going into detail. So just to sum the fMRI part up. So fMRI can probe function of the brain. It measures basically energy consumption by increase or decrease of oxygen. The most often used effect is the bold effect. You can do functional imaging differently, but bold effect is the bold fMRI is the method that is done in 90% of the cases. It's based on the fact that T2 star changes if there's oxygenation. We do not directly measure neurons. We measure the blood signal, a vascular signal, and this neurovascular coupling is very complicated because it involves blood flow, blood volume, and oxygen consumption. But it's highly correlated with the neuronal activity. And to measure it, we usually use a gradient EPI sequence, which is the most sensitive. And with that, I'm finally at the end. I have a list of very nice books or articles, 2 articles on diffusion, 2 books and 2 articles on fMRI, and well, 1 book, I think, and 2 articles on perfusion and fMRI, and 2 articles on review articles on SWI. With that, at the end, and I'm happy to answer questions.
Yeah, thank you, especially for the references, because it's really useful. We have 2 questions from the audience. So we can calculate the bold signal with both T2 and T2 star. If you want, you can see the question from the chat.
Okay, we start with the. You can.
Or I can read this.
Yes, yeah. So the first question is, can we calculate the bold signal with T2 and T2 star? Yes, that's right. That works. T2 star is much. The signal change in T2 star is much bigger. So you usually T2 star. If you look at the microscopic simulations, you find out that T2 actually gives you a smaller signal, which is originating from the capillaries, so more in the tissue bed, whereas T2 star is influenced from larger vessels as well. So in practice, it's usually done with T2 star because it's more sensitive, but T2 with spin echo may be more specific. So you can do both with gradient echo EPI or with spin echo EPI. But my advice would be to use gradient echo T2 star. Okay.
And before the second, sorry, so the spin echoes, for example, maybe can be used to explore other organs in which capillaries are more important. I don't know if you have experience of kidney or other outside from the brain. I don't know if you have experience, of course.
I would, well, not too much, but I would expect that. So T2 spin echo is helpful in the brain. In the other organs, I mean, because the capillary density is very high in the brain, as I showed in this micrographs there, I would expect that in other organs, in any case, it's better to use T2 star than T2. Okay, another question was SWI images show different contrast in different vendors on phase images, blood either white or black. Can you please explain the difference? Yeah, I wanted to get around that because this is complicated. So it depends. I think in the very basic MR lecture, you've heard that you go into a rotating frame. If you excite your magnetization, but it's precesses with the Lama frequency. Yeah, and the question is, does it precess clockwise or does it precess counterclockwise? And actually, of course, if you do Fourier transform MR, you have both senses of rotation, but you just look at one side, and this depends on how your scanner samples the phase. So basically how the phase shift of the scanner of the acquisition is done. And this is different between the different vendors. So they are basically clockwise and counterclockwise phases. And that was, if we go back, where's the SWI? Here. So Siemens, Canon, and also the Philips raw data, I think they are clockwise, and GE and Newsoft are counterclockwise, and also the Philips SWI protocol has this correction into this translation into the counterclockwise. So that's basically a method of how the electronics, the basic electronics, is adjusted by the vendor. But that's right. That's maybe very confusing because sometimes you have a bright and then the other time you have a dark contrast. But if you do pure SWI, it shouldn't be a problem. I mean, the advanced method for SWI is quantitative susceptibility mapping, and then you also get the sign, the positive or negative symbol. If you do the simple SWI, it should always be dark. But if you do the quantitative susceptibility imaging or mapping, then you will have this difference due to the different phase setting, clock or counterclockwise. But it's a very fundamental thing. That clear? Okay.
Yes, thank you. So we have no further questions from the audience, and I think you had had a great lecture, full of information. Maybe we have needed 2 hours to discuss all of them. So thank you for the lecture.
Okay, it was a pleasure. Thank you for moderating, and then have a great evening.
Yeah, see you next week.